welcome and thank you so much for joining us for this event. NeuroTech, by way of introduction, is the world's largest global nonprofit for the advancement of neurotechnology. And you are joining us tonight at Buzz in Review. And Buzz in Review is NeuroTech's biggest flagship event of the year, where we celebrate everything that happened in the previous year to kick off the new year that's just started. Tonight is the second night of our 3-week neurotech extravaganza across 3 of the final Tuesdays in January. We're celebrating everything that happened in neurotechnology in 2022, and tonight we are focusing on understanding the brain. To briefly introduce myself, my name is Sophie Valentine, and I am the London chapter lead for NeuroTech. And with me, you will see Cesar as well. Cesar, do you want to take a moment to introduce yourself?
Sure, thanks, Sophie. Hi, so I'm Cesar Echavarria. I'm a data scientist at Synchron, where we develop implants to turn thoughts into commands for digital devices. And very excited to share with you various updates from across the field of basic neuroscience research that have strong implications for the future of neurotechnology.
Awesome. Thanks, Cesar. So we're just going to give a couple more minutes. We do have a few people still filing in. So welcome back to people who are members of the NeuroTech community. We hope you've been enjoying all of the wonderful things that NeuroTech has to offer over the last year. And a really big welcome to anyone who's new and discovering the community for the first time. We hope your relationship with this community will be a long and fruitful one. Do hop over to our website, that's neurotech.com, to find out a little bit more about what we can offer. And you can even join our Slack community, which is where a lot of the action happens. So do introduce yourself in the chat, everyone, which hopefully is working. I know we had a couple of teething issues in our event last time around, but hopefully everyone is able to pop a quick message to let us know where you're dialing in from and what's brought you to this event. For those who joined us last week on the 17th, that was the first night of our 3-week neurotech extravaganza, and that was all about harnessing the brain. Don't worry if you did miss it. We will be sharing a link soon so that you can catch up on all of the neurotech goodness. And of course, we do have another night after this one. So in a week's time, we will be looking at enhancing the brain. For anyone who's in London, we do also have a watch party and networking event. So do check us out on sort of Twitter, Meetup, Slack, even find me on LinkedIn, as we would love you to join us for that networking event. But I think we are about ready to kick off tonight's event, which is all, as you can see, about understanding the brain. So we're going to have a recap of everything exciting that happened on this topic in 2022. So a big focus on research news and applications of research. And then we've got a wonderful talk from Dr. Dorian Haci, who is representing a brand new startup on the London scene. So really excited to have that later. And then, of course, we also have the NeuroBar. So if you've been to a NeuroBar event before, you'll know exactly the treat that's in store for you. And if you're a first timer, then brace yourself. It's great. So it looks as if we have enough people, and we are going to kick off our talk. And just before I hand over to Cesar, to kick us off with a recap of some things that happened in 2022, we want to take a moment to thank our global sponsors of this event. So Health Tech Connect, which creates the neurotech platform, the NeuroCatch platform. We'll be hearing a little bit more about them later too. And of course, the inimitable G-Tech. So Cesar, over to you.
Sure. Hi. So a lot of updates from this year. We have at the end of last year, we had the publication of this neurotech X primer, which is just a book that has a lot of information about getting started in neurotech. In April, we had the Paris chapter has had a booth in the Cognitive Sciences Forum, which is great. And then we also have in February we had a virtual digital therapeutics conference event showcasing innovative startups in the digital health space. And in May and June, we had NTX services releasing a comprehensive overview of the neurotech industry, which it's a great resource for people that are wondering, where is neurotech going? What sort of companies are out there what are the sorts of technologies that are up and coming. Next slide. Right, and in February we had the extended reality biosense, where we integrated neurotech and biosensors with immersive technologies. And we also had in April one of our newest chapters in Belgrade. They had a brain awareness event to kick off this chapter, and a lot of local interest in Eastern Europe. And in June we had the Brain Mind, where it was actually quite an interesting event, where just people were talking about, as we're developing technologies of various sorts, what are the neuroethical implications of these technologies, and how should we start tackling them, especially as some of the things that seem science fiction are starting to become reality. Next slide. Right, and if you haven't checked it out, I really recommend you to check out the NeuroTech Access Content Laboratory. This is basically a content initiative for the neurotech community. There's a lot of posts tackling all sorts of topics in neurotechnology, from technical sort of articles to more things that take a look at the big picture, see how different trends are coming up, what's the impact in society, and how to start regulating these types of industries. Also, they're always looking for writers and editors. So if this is something that if you have an idea for an article and a topic that you would like to cover, then you also want some guidance in how to go about this, you should definitely check it out. There's links on the Slack, on the neurotech X Slack, that will allow you to contact the organizers. Next slide. All right, so now we start with the research updates. As I mentioned before, there's just quite a lot of updates. We selected sort of like the ones that we think are going to have high impact in not just neurotechnology, but all sorts of technology. So, for example, this is one that caught our eye. So this is a study where researchers showed that individual neurons could learn by predicting future activity. This is relevant not just to basic neuroscience research, but it's also relevant to the future of machine learning and artificial intelligence. So if you're familiar with the technical details of the current state of the art deep learning, you know that this massive neural networks, the way they learn is they use a technique called backpropagation, where each neuron learns from the mistakes of the predictions that they made. This is currently not thought to be a biologically possible mechanism. So we think that that's not how it actually happens to the brain, although some people have some ideas of how that could be implemented in a biological organism. But anyways, what we think is a more reasonable way that neurons learn is to learn to predict the next inputs. So if you imagine, a neuron that's receiving input from the retina, from the sheet of neurons that collect light at the back of the eye, how will it learn that, what's the concept of objects? So one way that it could learn it is that it can notice that as a shadow passes over the eye, it's trying to predict what's going to be the next input. So if you have a shadow in one part of the eye, you sort of realize that you eventually learn that that predicts that there's going to be a shadow in the adjacent part of the eye. So by doing this, by predicting its inputs, it can also learn just some more general concepts like, okay, the fact that, points of light that are moving in the same direction tend to, form part of a cohesive whole, so they can learn about objects, all sorts of things. So this is thought to be like maybe one of the many reasons why biological organisms are just much more efficient at learning compared to like the current artificial intelligence systems that are out there, which need, just thousands of training examples. So this study that we chose to highlight shows that, kind of confirms that this is indeed how individual neurons are learning. They are learning to predict their next input, and it just it gives an experimental setup that now can be probed, and we can start learning some of the principles that happen in biological systems that we can translate to artificial systems that just potentially turbocharge the development of artificial intelligence. So next slide. Right. So you probably heard about this. This is pretty cool, and it's like straight out of sci-fi. So if you're familiar with the sci-fi novel Starfish that came out in the 90s, they describe this what they call head cheese. It's basically they call it head cheese colloquially, or neurogel more technically. Is basically a cultured brain on a slab. And in the novel, this is a type of technology that basically is interfacing with computers at such a high rate that it's just learning. It can learn skills at a very fast rate, faster than like humans can learn. And eventually, in the novel, this type of technology is used to basically have autonomous pilots of planes. So this is a small step towards that. So in this study, researchers actually from a company called Cortical Labs, they found that they were able to teach neurons in a petri dish to play Pong, right? So they received feedback about the state of whatever was shown on a screen, and they were able to then send out electrical signals that controlled a paddle in the game Pong. And through time, actually quite little time, I think it was about 5 minutes, they were able to complete the task pretty decently. So this is quite an amazing technology. And it's just a small step towards getting this sort of sci-fi technology out there. And this is yet another experimental setup that can fuel bio-inspired artificial intelligence, as we allows researchers to probe how such a simple preparation of nervous tissue, how they're able to learn just a very advanced skills. Next slide. Yeah, so this is also another interesting article that we chose to highlight. So as you may be aware, when we talk about brain-computer interfaces, we are typically thinking of interfacing with the brain using electrical signals, right? Using electrodes, injecting currents, but that's typically not. I mean, that's not the only way that neurons communicate with each other, right? In fact, the main way that they communicate with each other is through chemical means. So that's potentially another way to interact with neurons. So this team of researchers from China and Singapore developed an artificial neuron that can use dopamine for communication. So this is sort of the setup that they have here, or part of the setup. So this artificial neuron is made of graphene from a single sheet of carbons and a carbon nanotube electrode. They then added a sensor capable of detecting the presence of dopamine, and a device called memristor that is capable of releasing dopamine using a heat activated hydrogel attached to another part of the neuron. So what we're not seeing is basically the part of the neuron that is interfacing with the nervous tissue. They tried it out in both Petri dish of cells and a dissected nerve, and basically they found that this artificial neuron was able to detect dopamine, and then in reaction to those dopamine releases, then also interact with the nervous tissue by releasing its own dopamine. So this is potentially a new way that we can think about interfacing with the nervous system, not just injecting current, but interfacing with its various neurotransmitter systems, which are quite widespread and are responsible for a lot of different functions in the brain. Yeah, so next slide. Yeah, this is also pretty cool study. So back in March, the researcher from a trio of Swedish universities found, well, they think they found a better way to interact with the brain rather than the usual way of just putting electrodes in the brain. So as you may be aware, there are many incompatibilities between electronics and neurons. There's different material properties, but there's also one of the main ways that they differ is that they're relying on different electrical signals. So man-made electronic systems, they rely on the movement of electrons to create current, whereas nervous tissue uses the movement of ions for current. So these researchers created artificial neurons from organic semiconductors that are able to carry both electronic and ionic signals. And on top of that, there's the added benefit that this sort of materials are biocompatible and biodegradable. And they chose to test this idea out in a Venus flytrap because the mechanism that snaps the Venus flytrap is based on ionic conductance, similar to like the same mechanisms that control the activity within individual neurons. So they were able to successfully control that snapping mechanism of the plant, and they were also during their experiments, they were able to show evidence of Hebbian learning in their artificial neurons, which is Hebbian learning is this very core principle in neuroscience that neurons that fire together wire together. So they were able to see in their mechanical system, that's in their electrical organic semiconductor system, which was made up of multiple elements. They're able to find evidence of that type of learning that is usually observed in between neurons. So that's pretty promising and shows that this may be a feasible way for have a system that can seamlessly interact and process information from the brain. Next slide. Right. So this is a sort of now moving away from basic neuroscience research and looking towards the clinic. So a group of researchers found that they were able to ameliorate in a rodent model the symptoms of Parkinson's with a graft of neurons that produce dopamine into the brain. So just a little bit of background. So Parkinson's is characterized by the death of neurons that produce dopamine, and these neurons, the relevant neurons, are found in various regions throughout the brain. Some that are controlled movements, and that's one of the main symptoms of Parkinson's is the inability to control of losing the ability to control movement as well as a healthy individual. So this study demonstrates that neural grafts are able to effectively reverse these motor symptoms. So by putting the neuron producing neurons, the dopamine producing neurons in the brain of mice, they were able to reduce the symptoms. And these neurons were either induced pluripotent stem cells, which are adult blood cells that were reverted to the stem cell stage and then transferred back into the premanergic cells. This is potentially a therapy that that can be widespread and can provide a lot of relief for people that suffer from this disease. And I mean, this is the great news for people that have this in mind, is that this futuristic approach will soon be put to test in the first of its kind clinical trial in a specific population of Parkinson's disease at various sites across the US. So this is potentially something that will reach the clinic in the near future. It's definitely watch that space. Next slide. Right. So to finish with this section of research, just want to highlight this other very interesting research effort from UCSD professor Alison Muturi. So if you're not familiar with brain organoids, they're this self-organizing three-dimensional brain tissue that's derived from stem cells, and they're very useful because in a lab they allow researchers to study without the need of animal models a lot of different aspects of neural development and also disease. So one of the challenges working with this type of preparation is that, of course, if you want to study something like Alzheimer's, something that has a relatively late on stage, it takes a while for the tissue to reach the age at which some of the deficiencies of Alzheimer tissue becomes apparent, like the aggregation of tau protein and all those sort of traits. So what this group in UCSD is doing is they're piggybacking on the just the renaissance of space travel that has recently taken off, and they're sending these brain organoids to space. This took inspiration from reading research that the activity of human telomeres is altered after exposure to microgravity. This is just knowledge about decades of space travel and space medicine. So they sought to take advantage of this fact by then flying out those brain organoids for 6 months at a time, bringing them back to Earth. And in that scenario, they were able to see that this brain organoids actually basically hit the fast forward on the aging process. And so now in a short amount of time, you can have a very good model of disease in a petri dish, and that allows researchers to learn more about what's going on with Alzheimer's, what goes wrong, and also to potentially have high throughput screening of potential drugs, so that they can figure out what are possible treatments for this disease. So that this is actually quite the marriage between different branches of science. So I look forward to hearing more about this type of research. So now moving on to the next section. Talking about highlights from the world of human-inspired design. So first thing we want to highlight is this technology published in June by Caltech researchers, which is an artificial skin that can give robots the ability to sense temperature, pressure, and even toxic chemicals. Not just non-organic toxic chemicals, but even things like nerve agents or infectious pathogens, such as some virus that shall not be named. So the researchers sort of had various demonstrations for the technology. One of them, which is pictured here, with this with this researcher controlling a robotic arm, is one of the applications is remote sensing. So potentially there could be a robot arm that is in a scenario, in a setting where humans cannot access, but that still needs some dexterous movement, or it's a non-autonomous robot. So in this case, the human using NG could potentially control the robotic arm, while at the same time with this this sensing skin that they've developed, they can then basically sense the same factors that the robot is sensing. So maybe the robot sensing something like a toxic chemical, and that gets translated into touch for the user. So that's one of the possible scenarios, which is quite astounding. Another scenario is potentially developing autonomous robots. So with this sort of electronic skin, they can a robot can sense one of the target materials, and in that way learn to navigate or perform an action. So the researchers actually showed a proof of principle. They outfitted the boat with this type of sensing layer, and basically through the use of reinforcement learning, the boat was able to effectively navigate through a maze of hazardous waste. So a lot of potential promising applications from this type of technology. Next slide. Yeah, so this is sort of similar kind of technology. This is also an electronic skin. This skin, though, is made out of carbon nanomaterial, and the main purpose of it is to detect touch and heat sensitivity. So you can also imagine how this can be applied to the similar scenarios that the other polymer skin that we highlighted previously. It can be used for prosthetics, human-machine interfaces, self-autonomous robots that would allow that can use this sort of sensing to learn from the world and learn to act on the world. Next slide. So this is another type of stretchable skin. But I guess if you were paying attention in the other types of skins, one of the things you may have noticed is that they need a power source. So they're still sort of not very portable. You need to have them plugged in some kind. So this group of researchers at the University of Chicago developed basically an electronic, a skin-like electronic that can that has triboelectric nanogenerators. So these are small generators of the small components in the skin that are able to generate power from the user's motion. So this can potentially be used for something like long-term wearable device, which is what the researchers had in mind, because the sensors that are integrated into the skin, they are sensitive to various biomarkers that can be found in sweat. So yeah, Fitbit may become outdated in the near future as you are able to get even more information out of your body with seamless wearables such as this one. Next slide. So this is also another innovation on the realm of skin-like sensors. So these researchers at the University of Glasgow tackled the problem of large amounts of data being transmitted from these types of sensors. So if you transmit all the data from the sensors, you can have, to cover just your arm, you probably have like a couple of hundred sensors there. That's a lot of data to, process through and learn from. So these researchers from the University of Glasgow took inspiration from the peripheral system of animals, which as soon as it receives input on the skin, the peripheral nervous system begins processing at the point of contact, reducing it to only the vital information that needs to be sent to the brain. So in a similar fashion, these researchers built synaptic transistors. So they were able to do the processing of information within the skin itself, and then only transmit to a computer just the relevant information. So in that way, being able to reduce the information that computer needed to churn through to then get insights from this type of sensor. Next slide. Right. So this is actually quite cool. I mean, it's sort of not directly related to neurotechnology, but it's inspired by the human brain somewhat. So as I mentioned previously, there's a lot of sort of clunkiness to current artificial intelligence systems. They need a lot of training data. Also, the way that they're laid out on a chip makes them sort of cumbersome. You need like really big chips to be able to do fast learning and fast processing of information. So there's this type of computing called photonics, which basically takes advantage of the fact that you can perform computations with photons in much the same way that you can perform computations with electrons and having on-off switches. So you can have a light on and off, and the advantage of light is that it travels really fast. So using those two, using that to their advantage, they were able to build a system that can process information at the speed of light. And this may actually, it's an open question whether such an AI system can learn faster than biological systems, because right now the AI systems are limited by the fact that they need a lot of training examples, and then those training examples take a while to get churned through by our current technology. But potentially a photonic system may be able to churn those examples just at a much faster rate than even a human could, because of course, like there's a bottleneck in the communication between neurons. At the end of the day, that you're communicating between neurons through a gap, and you're relying on molecular diffusion. So there's only so much speed that you can get from a biological system, even if you figure out how to integrate a biological system with an electronic system, like we saw in some of those highlights. So potentially photonic system can just blow this out of the water and just be able to learn be able to learn from large amounts of data in a short amount of time. And definitely it's something to keep an eye on. It's very promising technology. And this is basically all the updates from human-inspired design, and I'm going to pass it off to Sophie now.
Amazing. Thank you so much for taking us through all of that. I never knew there were so many types of artificial skin. Just before we jump into our next section, we're at about the halfway point for our Neurotech X recap of everything that happened in 2022. We have had a couple of questions come in the chat. So do continue to share your questions with us. And so we'll come back and we'll answer those, or we'll answer them to the extent that we can at the end of the session. But thank you so much, Cesar. And now it is over to me to tell us a little bit about memory and cognition. So if I take us to our next slide. Okay, great. So what are we talking about here? We're talking about sport and we're talking about brain trauma. So the background of this is that we all know that sport carries risk. You could break a bone, you could tear a ligament, you could wear down your knees. But actually new research is drawing a direct connection between head trauma from sport and CTE, which stands for chronic traumatic encephalopathy. Got to say that one slowly. So this is research, a big research collaboration that's come out of leading universities worldwide. So Harvard, Boston, and Michigan in the US, Sydney and Melbourne in Australia, Auckland in New Zealand, Sao Paulo in Brazil, and representing the UK, Oxford Brookes University. And ultimately this analysis has drawn a kind of direct connection between CTE, which is a degenerative brain disease, and head impacts. So CTE specifically is something that causes progressive decline of memory and cognition. It can also lead to depression and suicidal behavior, as well as poor impulse control and aggressiveness. And eventually this is something that typically declines into a dementia that's similar to Alzheimer's, which, as we know, is one of the more devastating, both in terms of mortality and morbidity. We've got brain banks from the US Department of Defense, Boston University, Department of Veteran Affairs, and the Mayo Clinic. They've all published independent research showing this connection. But unfortunately, what we're seeing is that many global sporting organizations, including FIFA, for example, World Rugby, and the IOC, are all refusing to publicly acknowledge this relationship, obviously because it's an argument to be a little bit gentler when it comes to sport. But this is really, really significant, having a 68 increased risk of developing CTE, playing contact sports versus not playing contact sports. To put that into context, I think we all know that when COVID first became a global pandemic and we saw a really significant impact on age and took really significant steps to protect our parents and grandparents, that factor was in the single digits. So to see a factor that's as high as 68 really, really speaks to the severe impact of playing sport on this potential type of neurodegenerative disease. Next slide, we are talking about unwanted memory. So I'm sure we've all had a really embarrassing moment that we'd like to forget, whether that's, getting toilet paper on your shoe in the office, or, calling a teacher dad when you were a kid, or even, a mistake when you texted your ex. But we've got new research that's published in the Journal of Neuroscience, which was a collaboration between Chongqing University in China and Cambridge University, again, represent UK. And what this has showed is that we have a so-called guardian angel in the brain. So this is a system that basically proactively tries to identify unwanted thoughts associated with negative memories, and to minimize the impact those have on our conscious experience. And this circuit was made up of three brain structures. So the DACC, which stands for dorsal anterior cingulate cortex, as well as the hippocampus, which is really well known for its role in memory for any neuroscientists out there. And all of these brain signals work together to flag that intrusive memory, and to stop the retrieval of the memory and keep that out of our consciousness. And it's a process that happens below consciousness level, but really reacts a little bit more strongly if we go above consciousness. Obviously, the implications of this are huge in terms of treating conditions that are strongly related to negative memories. So things like PTSD, and can really help clinicians start to understand how we can continue to effectively treat these conditions. Okay, so now we are talking about a critical immune protein that has been identified as relevant in memory. So the background here is that our brains, in their normal day-to-day function, they stitch together distinct memories into one continuous narrative. And linking those distinct experiences not only gives us a sense of identity, but it also helps us learn how to survive and thrive in our environment. And so this research is all about what happens when that linking goes wrong. This research was published in Nature, so it's a big deal, and another research collaboration between American and Korean universities. And what this has done is identified a protein called CCR5 that's responsible for that sort of snipping memories and clipping them together. Established research shows that CCR5's levels rise during aging, and sometimes they can become a little bit overzealous, and that's what is one potential cause of memory loss in older age. But this study in particular found that using an FDA-approved drug for HIV treatment, so completely different to memory loss, they were able to restore an aging mind's ability to link memories in a mouse model. So the context here is that we have really significant implications for treatment of memory loss conditions, again, representing those minds that really struggle to snip apart and clip together those memories. Fun fact, CCR5 is a multitasker. As you can see here, it's something that's related to HIV, but it was also implicated in the 2018 case of a rogue scientist who used CRISPR technology to gene edit babies and was sent to jail. Pro tip, if you're going to gene edit babies, maybe don't use a protein that is involved in HIV, as well as memory loss and probably a few other things as well. So we now have some research from Scientific Reports Journal, and this is researchers from Kiel University in Germany. And what they were doing is they were looking at the sort of optimal working conditions for the brain as a way to understand how we can improve the design of modern computers. So brains are really, really efficient. I think the quote is that brains use about 25 watts of energy per day, which is pretty astounding if you think about how many times you have to charge your phone or your laptop, and how the tasks that phones and laptops do are maybe not quite as complicated as the power of a human brain. Of course, we know that really big AIs cost millions in computing energy bills to create and run. So if there's anything we can do to learn from the brain how to make efficient technology more efficient, then that's going to be a really big win for developers of novel technologies. And so what these researchers do is they created ultimately a model of the brain. So this was described as an artificial network of nonlinear oscillators. Anyone who's kind of worked in neuroscience before, the phrase oscillating will ring some bells for you. And they basically simulated ways to reach what was called a state of criticality. And that's really an optimum state where the brain is operating most efficiently. And they did that by changing the network's so-called environment. And this really makes sense, because if you think about it, as humans, we need to be able to really efficiently handle a variety of ever-changing environments to survive and thrive in our day-to-day lives. There was a wonderful quote from the authors, which was, to put it simply, one could say, our brain grows with its tasks even more than we had previously thought. But ultimately, this research is really useful as we continue to see more and more advanced artificially intelligent technologies. Finding ways to make sure that they're efficient as well as effective will make a real difference in terms of their adoption. So we have some further research that has come out of Texas A&M University and was published in Human Factors Journal. So this links a little bit to what Cesar was mentioning just now. And this is all about understanding trust in human-robot work interactions. So something we know is that as populations continue to grow, we're going to have more interactions needed than people to do the interactions. So if you think about online booking systems or elderly care, we've already started to turn to automation and the use of hardware or software robots. And technology advances are going to be necessary to facilitate these human-computer interactions, whether this is, a robot with a bow tie taking your dinner order through to a doctor interacting with a medical AI chatbot. And knowing how people are comfortable and how people prefer to interact with these technologies will have a really big impact in terms of their adoption. And so what this research showed us, they were looking into what creates trust in human-robot interactions. So the lab's human autonomy trust research has been focusing on understanding how operator states of fatigue due to robot collaborations and the robots making errors can cause them to trust or to not trust the robot. Really interestingly, this study used functional near-infrared spectroscopy, so FNIRS, for anyone who goes by the acronym. And this is basically a measure of how much oxygen is going to which parts of your brain, which represents how hard each part of your brain is working. And what they did is they connected particular brain regions to trust, and distrust was associated with increased activations in regions of the frontal motor and visual cortices, which basically indicated that the person collaborating with the robot in this task was having to work harder and harder and had heightened situational awareness as the robot continued to make mistakes, and that's what eroded the trust. The context here is that it's really useful to study these sort of things from a slightly more hardcore sciencey perspective than just asking people to report how much do you trust a robot. As obviously when you ask people things, it's going to be a bit impacted by bias. So what if you watched The Matrix last night? Maybe your brain wasn't working harder, but you might say you trusted the robots less when you were collaborating with them. And this research ultimately is useful to help manufacturers understand how to build trusted robots that people are willing and happy to use and can support growing populations. So this is my area of expertise, mental health. So I found this one really, really interesting. So we know that mental health is huge at the moment, for lack of a better term. The world is a bit of a depressing place in many ways at the moment, but also culturally, people are more open about mental health and seeking support for their mental health, which is fantastic. Everyone needs a therapist, as the old saying goes. But sometimes ways we assess people's mental health can be a little bit subjective and a bit fallacious because of that. So if we could find a way that was easy and accurate and accessible and could target early identification of mental health issues, that would have a really strong impact. If you think about it, a person having a depressive episode is least likely, that's the time when they're least likely to get themselves support. So if we have an objective, maybe even a passive measure, that can really help us understand when people need help. So this is research out of New York University's Tandon School of Engineering, and it was published in Computational Biology Journal. And what the researchers have done is they've developed a technology that measures a representation of brain activity by directly connecting to a person's skin using something called electrodermal activity, EDA. EDA is an electrical phenomenon of the skin that changes based on certain emotional stresses. So things like pain, exhaustion, even being rushed at work can impact someone's EDA. And usefully, it can be measured with fairly simple technology that could even be integrated into a wearable device. So Cesar, maybe Fitbit's kind of, as you say, going out of fashion, maybe Fitbit needs an EDA monitor. So there's a huge amount of potential for this technology to support the way that we monitor and treat mental health. Imagine having a wearable that could ping a friend or a doctor automatically to say, this person's having trouble, you should check in on them. But also this technology was identified as possibly helping with other conditions outside of mental health. So neuropathy, which is basically nerve damage that could arise for multiple reasons, for example, from diabetes, and can cause numbness, pain, or weakness. So the Human Brain Project, I hope everyone has heard of the Human Brain Project before. This is a really exciting project that ultimately aims to put in place cutting-edge research infrastructure that allows scientific and industrial researchers to advance what we know about neuroscience, computing, and brain-related medicine in general. Ultimately, they are a huge collaborative effort to create a computing infrastructure to facilitate research. And this is one of those examples of wonderful research coming out of the Human Brain Project that was published in Science Advances. So the background here, as mentioned earlier, is that the brain processes things in general, but in particular images, in a much more efficient way than modern artificial neural networks that software engineers and machine learning engineers can develop. So what scientists do is they try to take inspiration from neuroscience to create neural networks that function similarly to biological models, which are more energy efficient, so we can build artificial models that are really energy efficient. And what the researchers did here is they essentially created a model that aimed to represent the mouse visual cortex, which is the first part of the brain to receive and process visual information. And then they tested the model and found that it was able to respond just as they would expect a real mouse visual cortex to respond. So this is really exciting to be able to create a robustly brain-inspired artificial neural network that behaves in the same way as a real mouse brain. Again, it really leads to encouraging opportunities for advancement and adoption of artificial technologies, and particularly their accessibility. So any technology that can run on your phone rather than on a cloud-based server is going to be a little bit better from a privacy perspective, but no big machine learning model is going to run on any modern phone at the moment. So it's a step in a positive direction. Digital twins. Has anyone heard this buzzword? I think it was around in healthcare a lot last year, but actually I found out that it was initially launched in 2010, or initially introduced in 2010 as a term, and it's just becoming a little bit more prominent in healthcare at the moment. For anyone who hasn't heard of this buzzword, a digital twin is basically a virtual representation of real world things. And they're useful because we can do simulations and we can apply changes and we can monitor those digital twins and learn things that represent experiments we couldn't do in the real world. So one example is that we're probably not going to build an airplane and stick it right into a storm right away without doing some simulations first, because chances are it's going to break, and that's a very expensive way to do it. But what we can do is we can build a digital virtual model of the airplane and a digital virtual model of a storm, smush the two together and learn a little bit about what the next steps are we should take with developing our airplane. So the brain is probably a little bit more complicated than an airplane, which, you know, is saying something because airplanes aren't simple. But what these researchers are trying to do is to create a digital twin of a whole brain, which is a really, really exciting endeavor. So this is an EU-funded project called NeuroTwin, and it's ultimately aiming to design a computerized model of an individual patient's entire brain. And what they want to do is to be able to predict, specifically at the moment, they're focusing on how electromagnetic stimulation might support conditions like Alzheimer's disease and epilepsy. But ultimately, once you've got a digital twin of somebody's brain, there's a lot more testing you can do. And this sort of technology will really support personalized and predictive care, which are both huge themes at the moment in digital health. Both because as a personalized individual digital brain, you have your own digital replica. You're not generalizing to what everyone's brain has in common, but also predictive because you can start to simulate what happens if we introduce this drug or what happens if we introduce trauma and make predictions about how somebody's brain might behave in real life. And so fingers crossed, maybe this technology will start to turn those themes into reality within healthcare around neurodegenerative conditions. So I'm going to take a few moments now to talk us through some real world applications of research that we saw in the past year, because of course, what is research without an application? So first of all, we've seen an app that is able to identify Parkinson's and COVID-19 based on a user's voice, which is really, really cool and really relevant research. Voice is very big in digital health at the moment, mostly because it's a really, really rich signal. So you've got words, you've got pitch, you've got things like prosody. So what are the gaps people say even in between their words? And with both AI technology developing and kind of getting stronger and stronger every day, we've seen this with ChatGPT, as well as regulatory guidance really starting to shape up and help developers understand how they can develop and bring this technology to market. I think we can see a lot more of voice-related AI technology coming out in future. But this research in particular was from Royal Melbourne University of Technology in Australia. And what they did was they ultimately developed an artificial intelligence algorithm that was able to identify Parkinson's and COVID, as mentioned. They really focused on trying to use non-language sounds to overcome differences that people have in accent, which can really complicate and sort of add noise to the data. Interestingly, what they could also find is that they were able to differentiate between Parkinson's disease, a person with Parkinson's disease who was actively taking medication and a person who wasn't taking any medication. So one potential application of this is being able to monitor medication efficacy very, very sensitively versus blunter traditional tools that might survey someone's memory but not have the sensitivity to identify small changes. And particularly voice is very, very easy to capture, including remotely. This is something that maybe in 20 years we'll have it built into Alexa as an early warning sign for if someone is, you know, it's time to go in and have a conversation with your doctor and have more rigorous testing. The researchers themselves cited that more work needs to be done to bring this product to market. And I think we absolutely agree with that. You know, just as we've seen the issues over the past year with pulse oximeters based on hand size and skin pigment in both the US and the UK, a lot of technology like this, particularly AI, is really only as good as the data used to build and validate it. And voice in particular varies massively across loads of different demographics. So things like age, if someone goes through puberty or comes to older age, their voice will change. Things like health, if you have a cold, your voice can change. Things like language, if we have tonal languages like Mandarin versus something like English, there are really big differences there. Even socioeconomic status and ethnicity can cause changes in voice. So a technology like this really needs extensive validation to be really widely accessible and to avoid exacerbating existing health inequity. We also had some news from the FDA on ALS. So does anyone remember the ice bucket challenge from probably about 10 years ago now, something like that? That was all to do with raising money for ALS, which is, if I can pronounce it properly, amyotrophic lateral sclerosis. Again, another mouthful. And this is a rare neurological disease that primarily affects the nerve scales that are responsible for controlling voluntary muscle movement. So at the beginning of this condition, a person might experience twitches and cramps, which continues and progresses and can lead to things like difficulty swallowing and breathing. And very, very sadly, this condition is associated with mortality, often at around 3 to 5 years following diagnosis. So it's wonderful to see that we are having treatments approved by the FDA. And it was a really robust study, 24 weeks, multi-center, randomized, double-blind, placebo-controlled, all of these things you want to hear to have a good amount of trust that a study's results represent some underlying truth. But ultimately, the study demonstrated that the treatment group had a slower rate of decline on a clinical assessment of daily functioning than the control group. Clinical assessment of daily functioning would include things like how able is this person to do day-to-day tasks that allow them to function in their life. So things like making a phone call or preparing dinner for themselves. The context around this is really interesting. So as you can see on the slide, the FDA also gave this drug orphan drug designation. An orphan drug is a drug that treats a condition that affects fewer than 200,000 individuals in the US. And the reason we have this designation, which ultimately affords developers money to support the development, is these are drugs that are not super profitable and might otherwise not be developed. So it's really wonderful to see a proactive example of regulators stepping in, particularly given we're not particularly short of examples of sub-ethical choices being made independently on the part of big pharma. So I'm sure everyone has been watching the news and everything that's been happening with Alzheimer's over the past probably 2 years or so. We obviously had a little bit of a controversy with the first drug that was approved last year. That was a Biogen and Eisai drug that ultimately led to a lot of firings and a lot of investigations. But it's great to see that we have continued activity in the space around Alzheimer's disease, because, as mentioned, Alzheimer's is one of the most devastating diseases worldwide, both in terms of mortality and morbidity. And we'll only see an increase in the impact with aging populations. And of course, there really aren't any convincing cures yet. So it's wonderful to see the FDA starting to engage with this new drug from Eisai, which is lecanemab, similar to aducanumab, which was Biogen's first one, and starting to see positive results coming through for this drug, which is great. Although, of course, it's never quite as simple as that. We still have issues in terms of pricing of the drugs, of clinician confidence in prescribing them. So even when we do start to hear positive signals from regulators in this space, it doesn't necessarily mean that there's going to be a positive impact on patients. There are still a few more steps to take. And examples like this do tend to raise another question, which is, how should we be thinking about treatment for conditions like Alzheimer's disease? Should we be thinking about a reduction in cognitive decline, or should we start to be thinking about function or quality of life? Does it matter if you can perform better on a scale of cognitive ability if your quality of life isn't improved? The question is still out there. And I think this might be our last slide. So we also have mental health and brain performance monitoring collaboration between two companies, Kana Health. Kana Health is a digital health company that offers virtual care solutions, both technology and services, to providers and payers. And Total Brain, who was actually also recently acquired by Sonder Health, which offers mental health monitoring, as well as self-care activities and a way for patients to connect with clinicians. And we've got an integration between the two. So the Total Brains offerings are going to be integrated into Kana Health's platform, which will just increase the access and the number of people who can benefit from these technologies. So that's really, really exciting. And it's wonderful, as mentioned earlier, to see that mental health is such an important topic, and it's still a priority, particularly within the private space. We can probably expect to see more consolidation of digital health companies as we continue into the recession, as point solutions become a little bit less justifiable for patients and providers to adopt. But it's great to see a sort of targeted integration of mental health solutions into broader physical health solutions that Kana Health offers. So That is the end of our wrap up of everything that's happened in 2022. And of course, we do have a few more items for you. But what I want to do is invite Cesar to come back online, and we'll see if we can answer any of the questions that we've got in the chatbot. So I think, Vivek, can we get access to the recording slides? Absolutely. We'll be putting a recording live in the coming days and weeks. So do follow us on Twitter, LinkedIn, and you'll see everything promoted shortly. I can also see we've got a number of questions that have been answered in the chat. So thank you so much, Cesar, for answering those. And everyone on the call, do hop in and take a look at those answers. We've got a couple of open questions still. So from Anonymous, any advance on large array skin actuators stimulating transferring sensations such as pressure, temperature, touch, etc.? This one is not my area of expertise, but I don't know, Cesar, do you want to jump in? I don't know if it's yours.
No, not quite my area of expertise. Yeah, I'm not sure of any other type of large array skin sensor.
Yeah, it's a big question. Anonymous, what I'd say is join us in the Neuro Bar later. And apologies, myself and Cesar, this isn't our area of expertise, but I would be very surprised if there wasn't anyone in the Neuro Bar who could have a discussion with you on that one. And we've also got some input on Lynn on the Relivro drug that was approved. So thank you so much for sharing that, Lynn. What I'm going to do is just take a moment to tell you a tiny bit about a new technology that we have seen some advancement on this year. So I'm going to tell you a little bit about the NeuroCatch platform. That'll be 5 minutes or so, and then we're handing over to tonight's keynote speaker, which is Dr. Dorian Haci. So really, really excited to introduce him in just a moment. So Health Tech Connects, which is a company that creates the NeuroCatch platform, is one of tonight's sponsors. And we're really excited about their new product that has been released this year. And what I want to do is take a moment just to show you a video that will explain in much better words than I can a little bit about how their product works. So fingers crossed, our technology works okay. We've got 2 videos for you. I think they're only about a minute and a half each, and they'll just tell you a little bit more about the technology, how it works, and so hopefully you'll find this interesting.
How is your brain doing today? What if there was a device that could measure and monitor brain function and status like vital signs of the brain? That could be objective, accessible, rapid, and easy to use. Introducing the NeuroCatch platform. The NeuroCatch platform is disrupting the status quo for cognitive evaluation. Grounded in the science of brain vital signs, it can track subtle but significant changes in cognitive function by employing event-related potentials, or ERPs. ERPs are neurophysiological measures that are sensitive to changes in cognitive processing, which allows NeuroCatch to assess an individual's brain health across various states, conditions, and goals, such as brain injuries, neurological diseases, mental health, and brain optimization. NeuroCatch conducts a rapid 6-minute scan to measure and report on cognitive brain function. It produces a report immediately after the scan to display information on 3 different brain responses, or ERPs. These ERPs are A, auditory sensation, B, basic attention, and C, cognitive processing, termed the ABCs of NeuroCatch. The results of the scan provide a healthcare practitioner with objective measurements of these specific brain functions. These measurements can be used to help their clients identify areas for improvement, customize their treatment plan or journey to recovery, and understand how their brain function is today. NeuroCatch, objective cognitive evaluation, rapidly measuring brain health at the point of care.
So you can probably see why this is a technology that we were really excited to shout about. And I don't know if you're like me, but every time I hear about a new neurotechnology that's come out, my immediate question is, yes, but how does it actually work? And so this short video has got a few more details to answer that question. There we go. So hopefully that answered the question of, yes, but how does it actually work? What I'll do is just take us back to our main presentation here. There we go. And what we were keen to do is just take a moment to have a look at exciting developments that NeuroCatch, the NeuroCatch platform or Health Tech Connect have experienced this year. And you'll see why this is a cool technology that we're excited about. And for anyone like me who used to work in the EEG space, I'm sure you'll have recognized a lot of the kind of waveforms that we saw just now. So NeuroCatch at the moment is focusing on continuing support for their initial product, which was NeuroCatch 1.1. And amazingly, this has been used as a primary measure in a trial at the Mayo Clinic to identify a dietary supplement as protective against subconcussive impairments. As we've seen, trauma and sport, brain trauma and sport tend to go together pretty much hand in hand. So anything that can support individuals to continue to play sport without having significant negative impact on their cognitive abilities is definitely a win in our books. They've continued to integrate feedback and learnings about their platform into a new version of the technology, so that's NC 2.0, and done a lot of design and testing to continue to improve this product. And amazingly, they've also received 2 regulatory approvals. One is Health Canada approval to use this technology as an aid in diagnostics around cognitive conditions, but also FDA registration as a Class 2 medical device, which is no mean feat. Their team is also growing, so we can probably expect to see some more exciting things from Health Tech Connect and the NeuroCatch platform in coming years, months, days. And we do have just one more slide, and this is a recap of all of the exciting news that we've seen with the NeuroCatch platform this year. So we had a publication in, the team had a publication in Neurotech's Content Laboratory, which was their first product review, which is really exciting. We had Red Bull athletes who use this technology for functional assessments, also high school research and a TEDx talk, which is very, very cool. There was an award that was won around defense innovation, so kind of supporting defense in the US, a partnership with BCHL. I can't remember off the top of my head what BC stands for, but I think it ends with Hockey League, which is, again, pretty cool, seeing use of these technologies to support safe activity in sports. And expanding into pediatric assessments. So some really cool developments from NeuroCatch at the moment. So I'm really pleased now to hand over to our keynote speaker for tonight, which is Dr. Dorian Haci, who is co-founder and CEO of MintNeuro. So I'll let Dorian introduce himself, but I do want to lay a couple of lines down in case he's very humble and prepare to be intimidated. So he's a neurotech entrepreneur with vast experience in engineering and research, and he specializes in bringing really cutting-edge medical technologies and neural interfaces from the research lab into the clinic. As mentioned, he's co-founder and CEO of MintNeuro, which really excitingly is a brand new startup on the London neurotech scene. So personally very enjoyable for me as a Londoner. And this is a spin-out company from Imperial University. And in addition, Dorian is a translational researcher within the Next Generation Neural Interfaces Lab at Imperial and an Enterprise Fellow at the Royal Academy of Engineering. So I wasn't lying when I said it was time to be a bit intimidated. So what I'll do is stop sharing my screen and hand over to Dorian to tell us a bit more. And any questions you have for Dorian, pop them in the chat and we'll do a Q&A at the end.
Thank you very much, Sophie. I hope everyone can hear and see me. Well, that was a great introduction. Thanks a lot. And you made it easy for me to introduce myself because I was about to repeat the same things that you said. Let me just try to share my screen and let me know if you can actually see it.
Yes, we've got you. Thanks, Dorian.
Give me just 1 second. Okay. Oh. Yeah, so thanks for the introduction. Hi everyone, I'm Dorian, and today I'm going to talk about implantable neurotechnologies and their translation from the research lab to the clinic. Sophie made a great introduction, thanks for that. Just to repeat a few points, I'm the CEO and co-founder of MintNeuro, a newly formed spinout from Imperial College London. I'm an electronic engineer as a background, and for the past decade, I've been part of the Next Generation Neural Interfaces Lab at Imperial College London. And more recently, as Sophie mentioned, I've joined the Enterprise Hub at the Royal Academy of Engineering. At MintNeuro, we are redefining neurological interventions with implantable bioelectronics. We're proposing a new approach for developing brain implants that allow for a less invasive, yet more precise intervention. These devices are based on microsystem technology that allows to develop chip-scale devices the size of a peppercorn, even down to 1 millimeter in diameter. Many devices can be used to form a distributed platform of autonomous sensors. This enables the monitoring of a wider interfacing area and provides a multi-directional intervention tool. They don't require a battery, as they are wirelessly powered, and they communicate with the outside world through a wireless data communication link. By reducing the size and by removing wires, we are enabling implantation through a minimally invasive surgery compared to today's implantable devices. Moreover, we're taking advantage of the exponential growth of the semiconductor industry. Thus, we can leverage economies of scale, driving low-cost manufacturing and distribution. Our goal is really to reduce invasiveness as much as possible and to enable widespread adoption so that what was once considered last resort will progressively become elective at earlier stages in disease management. We have assembled a team of world-class experts in microelectronics, neuroscience, micropackaging and encapsulation, neural engineering, and medical devices. We have brought also medtech industry pedigree and clinical expertise. My talk was called From the Research Lab to Clinics. So I will try to show you my journey going from being a student to actually bringing technologies to clinic. And it all started about a decade ago in that lab up there. And I became part of the next generation neural interfaces lab, which aims at creating innovative neurotechnologies to enable communication between the nervous system and electronic devices. The two core directions of development of the technologies are research platforms for neuroscience and translational medical devices. The research team has grown and changed during the years since when I joined, and even during the pandemic time in 2021, we became also cartoons. One core aspect of our technologies is, sorry, I can't see. Yeah, so is developing neural chips. Meaning that we are developing microelectronics that enables medical devices. Yeah. During the years, we have moved from different generations of these neural chips, developing recording chips and stimulation chips, going from 1 to 8 channels in terms of neural stimulation, and from 16 to 32 channels in terms of neural recording. The project that I joined initially when I joined Imperial and the Next Generation Neural Interfaces Lab was the CANDU project, which was a multi-site cross-disciplinary project to create an implantable system to prevent seizure development for drug-resistant focal epilepsy. This was a collaboration between Imperial College London, University College London, and Newcastle University. The goal was to develop a new system implant concept that was based on 2 modules, one implanted in the chest and one implanted in the brain. Another strong feature is the presence of a novel closed-loop algorithm that will allow for the recorded signals from the brain to inform stimulation patterns. And stimulation was done through optogenetics. For those who don't know much about optogenetics, is a technique that allows to control the electrical behavior of individual neurons with optical stimulation. And that is achieved by genetically modifying the neurons, and then so that they can be turned on and off with light. This allows for high sensitivity, spatial resolution, and temporal resolution. We have developed various chips during the years, and what you can see on the right is the latest development of the CANDU 4 chip that allows to record from 32 channels and also to stimulate with 16 LEDs per shaft, as you can see there. Another project that we worked on during the years in the Next Generation Neural Interfaces Lab is the NGENI project that aimed at developing a next generation neural interfacing technology for brain-machine interfaces that is applicable both to central and peripheral nervous system. It uses similar approach to what we're doing in MintNeuro, which is a distributed approach with multiple devices freely floating in the brain, highly scalable, and multiple recording sites for each of the devices at different depths in the brain, in the cortex. Different prototypes have been developed during the years, both in terms of chips, in terms of encapsulation, but also we have focused as well in the implantation system. During the years, going from CANDO and NGENI, which were the project that led to the creation of the IP of MintNeuro, we have gone through various inflection points. And I just wanted to give you a bit of a taste of all those points during the year since when I joined to now, and then to introduce the next step in our journey. At first, we filed a patent on a BCI system. And in 2014, also the CANDO project started, which I mentioned was a collaborative project, but was funded by Wellcome Trust and EPSRC, a 10.5 million project. Then in 2015, our team, thanks to Tim Cosandino, who is the leader of the NGENI team, we received an EPSRC fellowship that supported underpinning research, and that led to the creation of the NGENI project. Then we secured more IP, and we developed the first wireless microchips that were designed and manufactured. Then the concept of our chip scale implants were introduced at the World Economic Forum in Dalian. In 2018, then our technology was featured in the UK Chief Medical Officer's annual report. In 2019, we presented at the NER conference. We published a conference paper and we presented the whole distributed chronically implantable platform system. In 2019, the team was selected as part of the MedTech Superconnector, which is an accelerating venture building program led by Imperial College London that allowed for both training in terms of entrepreneurship, but also for funding the development of the technology. In 2021, then MintNeuro, the venture, joined the Royal Academy of Engineering Enterprise Hub and their accelerator program. And that's where I spent about 1 year myself going through entrepreneurship training and startup training. Last year was the important year, let's say, because we managed to form officially MintNeuro Technologies Limited, MintNeuro, and to go through the spin-out process and to finalize it. The last point is what I'm revealing tonight, which is that we have officially won a grant, an NIHR Invention for Innovation grant, for developing a first-in-human, for a project that is developing a first-in-human device for clinical studies. And that is starting in January, has started. And it's a 1.4 million collaborative grant. So we went from the lab to the clinic, as I was mentioning at the beginning. So this project that I just mentioned is the way that we are getting into the clinic. It's a collaborative project between Imperial College London, King's College London, and MintNeuro, and is funded by the National Institute for Health and Care Research. I need to make an introduction to epilepsy. Some of you might be familiar with it, but just for everyone, epilepsy is a condition that affects around 600,000 people in the UK, with roughly 1/3 unable to control their seizures with medications, and subsequently being diagnosed with drug-resistant epilepsy. These patients have the solution besides the medicines is one of the best solutions is to go through surgery, really, and remove the spot in the brain that causes the seizure. This is highly invasive, as we know, but before going through the surgery, there is a period called the preoperative neuromonitoring period, in which patients are being monitored initially with MRI and EEG devices. Then, if it's not possible to identify the focus in the brain that causes the seizures, they have to go through implantable neuromonitoring systems, often through ECOG, so electrocorticography, or through SEEG, stereoelectroencephalography. And this implantable period is done in the clinic and is done through implanted electrodes that go through the skull, go through the skin, and they connect to what clinicians call the wall, which is the system that records signals in real time and over time. And that is a process that takes sometimes up to 3 weeks because they don't manage to identify the seizure. And that's done before the surgery. So as you can see, there's many limitations and drawbacks of this technology because of the distressing period that patients have to go through, and because half of the time, actually, patients are not, they don't go through the surgery to remove the spot because clinicians haven't managed to identify the right spot for surgery. What we are developing, what projects aim to develop, is a wireless intracranial neuromonitoring device that addresses these challenges by adopting a proprietary microchip technology provided by MintNeuro. This technology will make the devices small, portable, and wireless, while ensuring that the data quality is not compromised. This will pave the way for future devices that will require only a minimally invasive procedure, making the technology available to a much larger patient population. The team's vision is really to create a minimally invasive device that will allow patients to return home soon after the implantation surgery. This will allow for a longer monitoring period, even more than those 3 weeks that patients go through in the clinics, with then patients being free to continue their normal lives, leading to less patient distress, more seizure captured, better localization data, reduced clinical cost, and ultimately better surgical outcomes, resulting in a substantial long-term cost saving as well for the NHS in this case. Now I wanted to give you some of the challenges of developing opportunity for implantable neurotechnologies. Just quickly, because I don't think I have too much time, but just wanted to highlight that about a couple of years ago, I gave a talk on actually the challenges and opportunity for implantable medical devices and neurotechnologies within the NeuroTech X community. That is on the NeuroTech X channel. And I go in depth on the actual technology limitations and the opportunities for these devices. Moreover, I wanted to highlight that in 2019, the Royal Society, which is a UK national academy, published a report called A Human Perspective, Neural Interfaces. This report highlights 2 challenges. The first one is to fulfill the potential of neural interfaces in medicine and to manage the risk associated with the wider societal use of neural interfaces in everyday life as they emerge from specialized markets and become available to consumers. This was done as a call to action for government, academia, and industry. Moreover, since we're in the UK, in 2020, Innovate UK from UKRI established something called the Knowledge Transfer Network on neurotechnology. And in 2021, the Knowledge Transfer Network published a report called a Transformative Roadmap for Neurotechnology in the UK. The document aims at providing a clear roadmap for development and translation of neurotechnology and BCIs in the UK. Again, this is also a call to action directed to governments, universities, academia, and industry as well, to establish a collaborative mechanism that will accelerate development of meaningful and highly effective neurotechnology. Next slide is my last slide, and usually I always try to end up in a positive end. However, what I'll do today is to go to the first slide that I usually use when I have pitch decks, which is going to the problem, because I want everyone to go back to the reason why we're developing these devices and we're going through long processes of creating ventures and projects and asking for grants and investment, is really to solve the biggest challenges in treating and managing neurological conditions. That's it. Thank you.
Thank you so much, Dorian. That was really, really fascinating, and absolutely, I would also recommend reading both of those documents that you shared. I think really, really insightful, particularly for, again, UK represent. But it's great to see people starting to put on paper, these are the topics we should be thinking about. So we've got a quick Q&A, and thank you to everyone who's been sending questions over on Slack. I think we haven't had any come through just yet in Zoom, but do keep. Oh no, sorry, we do have a couple, from anonymous questions for you, Dorian. At what stage MintNeuro wireless implant? So how advanced are your experiments at the moment? So are you doing sort of in vivo in animals, or I think you mentioned first in human is coming this year. How far have you got on that front?
So it really depends on the on what we're referring to, because during the years since 2012, actually, there have been development of the chips, the neural chips that I showed earlier, and those have been tested in both in benchtop system, but also in animal models. And we have a collaboration we've had in the past with our NGNI lab, and we continue to have a collaboration with Newcastle University, where there's an animal facility. And also some of the devices have been tested in animal models and also in macaques, so non-human primates.
Awesome. Yes, thank you for that detail. We've got another question around the fundamental driver for this project and whether it's a tech-driven or a needs-driven. So did you sit down one day and say, we really need minimally invasive implants? Or did you stumble across a technology that you then realized could be used for that? And so how did you work out the marrying of the need and the technology?
So there's in the team, we're really from various backgrounds. And so in my case, I have an electronic engineering background. So I started from the technology perspective, so engineering perspective. And I realized that the technologies that we were developing within our research lab were really advanced and cutting edge technologies. And also comparing with other groups and companies in the world, we were up there in terms of the quality of the technology we're developing. For that reason, then I focused more on finding different directions for using our technology for creating more impact for neurological conditions, which met also with my passion for neuroscience and neurotechnology. And but in the team, we have people and it really, we brought together the team coming from very different directions. And but in my case specifically was to use technology to solve critical problems in the world, specifically for neurological conditions.
Yeah, absolutely. I think there's definitely a need there, and to see so much opportunity from the tech side is really incredible. And a couple of things that you mentioned in that response actually tie into another question we've got here on Zoom and some of the questions coming in on Slack. So Jake has asked, what kind of competitor technologies are you aware of? And that question dovetails nicely with a question we had come in on Slack, which was, how do you see a technology like MintNeuro's comparing to something like Synchron or Neuralink or BlackRock?
Yeah, very good question. And I'll refer actually to the talk that I gave last year to NeuroTech India, which was, I went quite in depth of the technologies that are out there. Synchron, I spoke about Synchron, I spoke about Paradromics, Neuralink, BlackRock, which have been around for a long time. everyone has their own different approach. Some come more from a technology perspective, some come more from a clinical perspective, and so I think we are all so. Often people try to use or like mentioning competition, which when it comes to really companies, often is about competition. But I think in the landscape where today of neurotechnology, we're all collaborators and partners. I mean, we speak to people, Paradromics, I have contacts with them, I have contacts with BlackRock and others. And so it's really about helping each other to really move the field forward. And in terms of, going back to the question, in terms of comparison with the with other companies and other technologies out there, it really depends on where the focus is. Some focus more on accuracy of the recording. Some focus more on the patient response and how patients feel with the technology independently on the accuracy of the technology. Some focus more on the number of channels or electrodes. And it really depends where we're heading. So what we're doing with MintNeuro is really trying to reduce as much as possible the invasiveness. That's one of the things that I was mentioning earlier, and to really make what was once considered last resort something that can be used at early stage in disease management and treatment. And those then inform the technology features that we have.
Yeah, absolutely. I think you're right. The conversation always finds itself turning to competition, but we had it, I think, summarized as well really nicely in our last week's event, where both of our speakers said, this is not going to be a winner takes all game. There are so many complementary technologies and there are so many conditions that need support that, competition is the wrong question, and it's more collaboration is what we should be thinking about. So I think we've got time for maybe just a couple of more questions. And there's one that came in on the technical side, which was whether your technology, either now or in future, might have the ability to stimulate nervous tissue.
Very good question, because we're focusing on sensing at the moment, and the project for epilepsy is focusing primarily on sensing. However, the main vision in the long term is to having enough data that will inform, and that's what we mean by redefining neurological intervention, will inform the future of intervention, meaning that could be support both for example, stimulation. So stimulation could be added because we have also experience in developing chips that stimulate, but also in terms of informing medical interventions, in terms of medicines and drugs to be taken by the patients, or other forms of intervention, or even just an alert. Sometimes in epilepsies, it's important to know when a seizure is about to happen and to predict that in time from happening. And so that's what really we would like to do. We would like to apply this technology initially for sensing, and then to move to informing different interventions. And that could be also by closing the loop in real time and stimulating, like some other technologies in the field are aiming at doing.
Yeah, absolutely. I think that totally makes sense. You definitely don't need to stimulate to inform sort of interventions or to support a particular condition. And so I think our final question, and a few different versions of this question came in, but it's really about the major challenges you had in the translation process. So I know you mentioned you went through some accelerators and you did some entrepreneur training. For you, what was the toughest hurdle in the translation process, whether that was product development or testing or regulation or even just running a business?
I guess more than a hurdle, I think the thing that I spent most time on is really developing the vision and the team and trying to bring together the best of the best. Might sound cheesy now, but the idea was to bring together a team with different backgrounds, coming from electronic perspective, as in my case, but then also scientists, because we need neuroscience to work, and also to have the vision of clinicians and to know how these technologies are going to be used then in the future in clinics. So I would say, in my case, I think it's quite key to bring together the the team. Then from a challenge perspective, from technology perspective, I think regulatory is a very big issue, but I mean issue for kind of like translating these technologies from a company perspective, from research perspective, but it's good that there are some rules in there to kind of like make sure that everything works fine. But that has been a challenge, especially in Europe in the past few years with introduction of MDR in EU, but then in the UK going back to what it was before. So there are some challenges in there, and I think that's one also of the key challenges. So regulatory. Then from a technology perspective is about really bringing together electronics and kind of like on one end having high accuracy and very cutting edge technologies from an electronics perspective, but also encapsulation and packaging, because you need these devices to stay in the brain for a long time and to be really reliable in time. Yeah,
absolutely. That
I can go on for hours to talk about challenges, because that's really I think many people spend more time on challenges than anything else.
Yes, absolutely. But I love that you included in your answer, one important thing that could be a challenge, but is really empowering, which is bringing in great people. And I think, it's true, that old adage that you're only as good as the people, and without people, you have nothing. So really wonderful to hear that. And certainly, I think regulation, it's a very changing landscape at the moment. It's quite fractionated geographically. So certainly a challenge there. But Dorian, thank you so much for sharing your journey and your insights and answering all of these questions. We really appreciate hearing from you. So we'll wrap up your talk there, and thank you so much for joining us.
Thank you, Sophie.
Thank you. And just before we wrap up this evening, we do have one final speaker who's a semi-surprise speaker, someone we were hoping would be able to join us tonight. And we have, while Dorian was speaking, just received confirmation that Guillem Monso will be able to join us and briefly tell us about their research on brain-on-chip technology to better understand neural circuits for medical and computational applications. He's also a lead for neurotech Barcelona. So thank you so much for joining us, Guillem, and I will hand the mic over to you.
Hello, can you hear me well?
Yes, we can.
Fantastic. I'm going to share my screen. Good. Can you see it?
Yes, yeah.
All good. All right, so hello everyone, this is Guillem Monso. I hope the top part doesn't disturb you. In any case, I'm going to introduce you the topic of today, which is basically, I'm going to discuss a topic related to the use of brain on chip in order to better understand how to understand neural circuits for medical and computational applications. In this case, I'm going to describe a project which is related with my research group. This is not a research that I did of myself, but I think it's interesting to go through it. My name is Guillem Monso. I did these degrees basically. I did my associate degree on control, and finally I did my master's in neural engineering, and now I'm pursuing my PhD on brain-on-chip technology. Before that, along these years, I've been doing different projects. At the beginning, I was working more in the nuclear and particle physics field, which I was working at the ALBA particle accelerator. Later on, I moved at the ITER fusion nuclear facility. And finally, I work after my master's in neural engineering. I did basically the design and simulation and analysis of a vagus nerve neurostimulator for in-brain neuroelectronics in Barcelona. As mentioned, I'm going to talk to you about this project that you can see here. And well, basically, we're going to go through it. Here, basically, what you need to understand is that brain-on-chip technology basically is it's growing neural cultures on microfluidic or microelectrode arrays. And well, there are different approaches, of course. One of them is doing like 2D cultures, as you can see on the right. But basically, these also might disturb the structure of the neural circuits. That's why there are other approaches in which they basically grow the neural circuits as brain organoids, for example. In this case, this might not be considered as a brain organoid itself, 3D structure. Some of the advantages is basically it enables to get a closer look, closer network. We get a basic a network structure which is closer or one of a in vivo case, and yeah, it improves the expression of differentiated function and tissue organization. This might then help us to better understand brain diseases and also reduce animal studies. But basically here, what we in order to determine and study this network, we've basically placed a microelectrode array, which is basically a set of very small conductors, let's say, that they are connected into this machine that you can see here. This would be the plate on which we grow the microelectrode, the neurons, sorry. And well, why using the microelectrode array? Well, because fluorescent-based characterization is it's static and often cytotoxic, and which, yeah, it doesn't enable to study the neural activity for a longer periods of time. And well, it basically it's not level free in situ assays for continuous readouts. Here in this study, basically, are proposed two different types of containers in which the neurons are grown. In the bottom case, basically, it's a simple cylinder, let's say, donut shape, which basically the neurons are grown in the middle, and the media, which is the liquid which provides with nutrients and oxygen and so on to the neurons, it's supplied on top. However, there is this proposed design which basically supplies media through a membrane, porous membrane. And well, the idea is that this design might help into the supplying nutrients and oxygen into the neurons. And this study basically goes through makes this comparison. Here you could see there's basically the design and construction of this bioreactor. And well, this would be placed on the middle of this microelectrode array for the readings, the readouts of the neural activity. Some other step is basically the obtaining the neural networks. Something that is very important to consider is that until now it was very ethically complicated, since neurons were often and are still often obtained from primary sources, which is basically from animal and embryos. However, now with different advances in biotechnology, it's possible to obtain neural tissue and cells basically from the differentiation of human iPSCs, which is pluripotent stem cells. And once we obtain these cells, which basically later on differentiate them into neurons or other type of tissue. But in this case, we are interested in neurons. In order to obtain the 3D structure, then what we do is we mix this neural with this iPSCs with hydrogel. And well, basically, once these cells or iPSCs are distributed in a 3D environment, as they get to differentiate, they can grow their axons and get to connect with each other in a 3D structure way. Here you can see the different layers of the same 3D body from a neural structure, and basically, yeah, it's possible to cut them with a microscope and get a picture at different layers from the same organoids. And well, after we have outgrown these neurons on the microelectrode array that you can see here, it's possible to basically, through this device, acquire the data in order to later on make the post-processing that you can see here. This would enable us to basically perform this statistical analysis. On the left would be the one obtained from the microbioreactor, and the one on the right is from the group 2, which is the simple PDMS ring. Here we can, from a simple look, we can see that there is a difference in performance on the neural networks, let's say, since, for example, the inverse spike ratio and the numbers of spikes and so on are much lower in the right side than on the left side. So because of this difference in spikes and so on, it was possible to determine that the microbioreactor might provide advantages on neuron survivability, and therefore it might be more appropriate to sustain 3D neural cultures in the longer term. So yeah, basically as conclusions, we can say that some of the insights were that the neural culture matures and neuron spikes organize as neural culture matures, neuron spikes organize in bursts instead of occurring randomly. And basically the number and duration of bursts were higher in the microbioreactor, making it more appropriate for 3D neural cultures as increases nutrient and waste exchange. And that's it. Thank you very much for your time. And yeah, I think it was time appropriate. I didn't want to extend it more than necessary. I hope you liked it.
It was, absolutely. Thank you so much for going through that and kind of telling us all about your work. Again, really, really fascinating and such a compliment to what Dorian was talking about. We have had a couple of questions come in over Slack, which I think we have a few minutes to run through quickly. And please feel free to stop sharing your screen. Hopefully it pops up. Guillem. Oh, no worries. Well, no worries. We'll see if we can put you off in a second, if not. So one of the questions that came through was, could you tell us a bit more about the sort of major uses or applications of your research? What would be the next steps?
Yes, currently, well, basically now we are working is better to basically get a much higher resolution on neural readouts. So, for example, we are reading a lot of neural activity, but in the same way that when you read EEGs, you read from like scalp EEGs and so on, you read from full networks of neurons. We are exploring more the possibility of reading like specific neurons in a whole network. So yeah, this would be the approach I am working on now. And yeah, as applications, for example, in the medical side, something that is a project enables you to do is basically there are cases in which people have like gene mutations, let's say, and because of that, they develop. Because of that gene mutation, they develop different neural diseases. And in order to better understand them, it would be possible to extract skin cells from the person. And when you extract these skin cells, you can then convert them into iPSCs, the stem cells that you are stem cells from that person. And after that, you can differentiate those stem cells into neurons. And from that, you can grow a neural network and study that specific, not the neural networks that are within the person, because those you cannot extract, but you can study how the different how the neuron cells behave when forming a network on a petri dish.
That's so fascinating. Yeah, really, really interesting. Thank you for sharing that. And one of the other questions that came in was, so if someone wanted to follow in your footsteps and do similar research to what you're doing, what are the major skills that you need to have to do your sort of research? Is it coding? Is it knowledge about neuroscience? What's important?
Yeah, I personally come from electrical engineering side, so yeah, something that I realized is that, well, along my years, I've interviewed different professors. Many of them were biologists, neuroscientists, and all of them say like an engineer can learn biology, but a biologist cannot learn engineering. It's a much, of course, everyone can learn everything if you have infinite time and resources. But as you grow older, the harder it is to switch careers, let's say, or to learn certain skills. So the skills that you might learn in biology, one could say, and I've not pursued a biology path, let's say, one could say that it's easier to learn biology than to learn engineering. And because of that, I would recommend to follow more of an engineering path.
Yeah.
Personally, I like a lot electrical engineering because you can basically, you get a broad application and set of perspectives. And yeah, coding definitely not, I would not recommend because it's not as hands-on, I would say, although it's interesting because it teaches you how to think. But with now ChatGPT and all these technologies, I don't think that software engineering is the best. Coding, like apply it on coding, is the best part.
Yeah.
But yeah, definitely electrical, I would recommend. But that's where I come from, so maybe someone else would recommend. Now I know that there are biotechnology careers and so on. So.
Mm-hmm. Yeah, absolutely. No, I think it's really interesting the balance between bringing all of the different skills and knowledge areas together in yourself versus like what Dorian was saying, working with a really multidisciplinary team. And just time for one final question that's come in the past couple of minutes from Anonymous. So thank you, Anonymous, for your question. And this was, are there any exciting companies doing things in the sort of research space that you would recommend looking into?
Companies working in this area.
Yeah.
Yes. I started with it because of this company in the US. It's called Koniku. So K-O-N-E-K-U. I can write it on the chat if you want.
Thanks.
Yeah, these ones, it's not applied on medicine or computation or anything. It just it's applied on smell, and because they create smell sensors from creating kind of cultures, neural cultures encapsulated in some small chip, let's say, and then they basically put these chips on strategic locations, let's say airports, for example, so they can detect smells and certain fragrances or odors that are liberated from, for example, explosives, drugs, or anything. And they already got a project with, they got funding from Airbus, which is one of the biggest companies in the aerospace field, and they are going fast, these people. It's very interesting what they are doing.
Awesome. That's really cool. Thank you for sharing the name as well. So that's just in the chat and certainly one for us to watch. Guillem, thank you so much for telling us all about your research and answering our questions. Really appreciate it. Really, really fascinating. Awesome. And then so what I'll do is I'll just grab the mic back just as a final formal wrap up of our day 2 today. So thank you everybody who joined us and heard all about our news. A couple of final notices before we let you go so as not to miss out on the best bits. There we go. So do join us next Tuesday for our final Neurotech Tuesday. That's our final event of Buzz and Review 2023, when we'll be talking all about enhancing the brain. So as we had today, we're going to have a wrap up of all of the wonderful things that happened under the umbrella of enhancing the brain in 2022. So we'll be digging into research, we'll be digging into what companies have been up to. But we're also so excited to have a really fantastic keynote speaker, which is Dr. Francis X. Shen, who's from the Center of Bioethics and Department of Global Health and Social Medicine at Harvard Medical School. Pretty cool space to be joining us from. And is also at the Department of Psychiatry at Massachusetts General Hospital, and is a Harvard University affiliated professor at Harvard Law School, and he'll be talking all about neuroethics, which, as we know, is becoming a really hot topic. So please do join us. Absolutely don't miss out. And again, a shameless plug, if you are in London, we are having an in-person watch party and talk and networking event in central London in Fitzrovia. So do join us at that, and you can find links to sign up to that event and find out more on our Twitter, Meetup, find me on LinkedIn, Slack, pretty much anywhere, but would love if you join us. So the final event of tonight is the NeuroBar. And as mentioned before, the NeuroBar is a fun social virtual networking event. I won't say anything about how debauched it's become in past, but we'll see what happens tonight. So what we're going to do is we're going to share a link to join the NeuroBar in the chat. We're also going to share it on Slack for those who are on Slack. And we'll be kicking off in just a few minutes at 3 p.m. EST, 8 p.m. GMT, so just kind of 5 minutes or so. So do click into the link and it will go live in just a couple of minutes. So NeuroBar link to come. If you're joining us tomorrow, please do. Sorry, a week from now, please do join us. And that'll be the same link that you joined for tonight's session. So thank you, everyone. Really appreciate you joining us. Thank you, Guillem and Cesar and Dorian, for all of the fascinating insights you shared. And we hope to see you in the NeuroBar in just a few minutes.